as Elsa announced, I will tackle the textile question like from another perspective, namely how we can have a deeper knowledge, a better knowledge of the textile we are studying when we collaborate with chemists. So I will talk about chemical analysis. Very briefly, for those who do not work with textiles, why it is important, like the slide, the smallest textile fragment, there's a set of many informations because it informs you about the resources which were available to the people who produce it. You can have a better knowledge of the technology they mastered, and which includes not only different weaving techniques and which implies the constructions of looms, but also, as you can see here on that colored fragments from Castrobrim, the master of dyeing activity. And of course, the context in which those textiles are found is an important feature, as it informs you more about the society. So who can, I mean, a person had to produce this textile for a purpose or for a person or for herself. So it's when we add the information from all these categories, we can better understand textile production and its consumption within past societies. And when we, like textile archaeologists, if I may say, we work with textile. So our first approach is a visual examination. And I will show you just in a few examples in a minute that especially in the Nile Valley, when we have such exceptional environmental conditions, where these textiles are often very well preserved. So with this, I may say, with simple visual examination, we can assess if a fragment is decorated or not. The colors are usually very well preserved, so it's easy to see if we have dyed threads or textile entirely dyed. We can, of course, identify the wave, and even with the naked eye, when you are used to your material, you can easily identify the fiber, if it is wool or cotton in our case. And of course, basing on all these technical features, we can have a preliminary assessment of the fact if the textile we are examining is a local project or if perhaps it was produced abroad and imported to the area. And so in this cases, in these bullet points, I also listed you the various steps we can work with. I mean, we can so examine this textile on various, I would say, magnifications. We can go deeper and deeper. And so, for example, if we have a look at this first category, decor dyes. So as I just told you, visual examination is with a quick look, we can see if this textile is decorated and or dyed. If we want to have a preliminary idea of the fact if it was dyed with plant or animal dyes, for example, we can apply such a technique as electron microscopy or spectroscopy. So, for example, this one first, which can, so it is a non-invasive technique, it's a portative technique, and with this method you can, for example, make the difference between animal and plant dyes, but it will not give you the precise source. If you use electron microscopy, it can be very helpful to know more about the composition of the mordant which was applied, because as I will show you in a moment, some dyes will need a mordant to be applied before dye your textile. But again, so these are non-invasive techniques and you will have some results, but if you want to know the specific source dye which was applied, you will need to recur to another kind of method, which are chemical analyses. And these methods are destructive. So this is like the minus point, but on the other hand, you can have more information about the composition of the dye source. So for almost each of this category, when we have technical features, we can apprehend them from different levels. So just to show you a few examples of visual examination. So here we have two cotton fragments from Odongola, which I studied 2 seasons ago. And you can see with the naked eye that the fragment on the top is undecorated, while the one below has blue thin bands. Here you have on the top fragment of half silk from Jebel Ada. And you can see with the naked eye the colors and the fragmentary decor, like vegetal kind. And you can see in the lower corner wool decorated with also colored and dyed woolen threads. You can also see the various disposition and width of bands. And you can see on that images that even with the naked eye, you can appreciate different width of the threads. And so these are things you can really assess with no specific material. But you can also, which is something which is really helpful on the field, you can use optical microscopy. So usually I worked with, now I think most of you know this kind of big pen dynolite microscope, which are just linked to your computer, and it gives you immediately an image at different magnifications. And as we are talking about dyes, as John Peter observed, you can see, of course, because there is the mechanical use of the fibers, so that it's like your blue jeans. You can see that when you wear them after a long time, there are areas where the blue is just has gone. It's because of this mechanical friction. So that's the first possibility. But also, as you can see on that fragment, I don't know if you can see my pointer or not. So in the lower left corner, you can see like the general view of the textile. So even with the naked eye, you can see that some threads are deeper blue, while others are maybe greenish or yellowish. So it means that we have this like using of the thread, but also from the beginning, perhaps those threads had different shades of blue. And what is really interesting, I think, when you use optical microscopy and you look deeper in the thread, you can see that the concentration of dye can really be very variable on the fibers of one thread, which is an important thing you keep in mind when you are sampling it for dye identification, because our eyes see just a blue thread, but in fact, the fibers retain very different amounts of the blue dye. So it's important to select a thread where this amount of dye is really well concentrated, will enhance the results of the analysis. Just before I go, I speak more about those dye analysis, which is also important is to know before you start, so how the dyes do work and also how the chemists work. So you will not be disappointed when you will have answers to your questions. So very quickly, we are talking about natural dyes. So we can distinguish two categories between plant dyes and animal dyes. And some dyes, such as henna, for example, can be applied directly on your fiber, but others such need the applying of a mordant. So, for example, madder is this kind of plant dye. So, just to show you very quickly, so on your left, the textile has been covered with several like mixtures of different mordants, of iron and or alum. And then this textile was dipped in a cochineal bath. And you can see that so the choice of the dye has a deep influence of the final color you will get. And also what is I think important to remember with that image is that what we see with our eyes is red or gray or black doesn't mean that it was dyed with a black dye source. Like you can see that the dye here was cochineal, which gives usually red color, and it's used as usually for that. But I mean, what our eye sees is not necessarily informative of the real components of the dye, so that's important. So VAT dyes, so these are dyes that are not soluble in water, so indigo tin, and so all indigo plants, and as John Peter stated, it is usually difficult, so this is something you must be aware that when we have the result of your analysis, it will give you the composition, and you will have several candidates to choose between. So it's not like you have one specific precise answer to your question. So how these VAT dyes work, you can see that so the indigo cannot be dissolved in water. So you need an alkaline solution, then you put your textile in, and you see on the left image that your textile is not blue in the solution. It's like yellowish green, greenish yellow. And it's at the moment when you take the textile from this solution that it interacts with oxygen, which will like clip your dye onto the textile. And with the mordants, you can, with the mordant dyeing technique, it's the mordant which will influence the final hue of your color. But with indigo dyeing, it's the number of baths which will make your textile, your thread, deeper blue, and you can even reach black colors. So how can you work with dye identification? So it's an assemblage of two methods. So HPLC is for high performance liquid chromatography, and chromatography is used to separate the various elements contained in your sample. And it's coupled with mass spectrometry. So the mass spectrometer is a very sensitive detector, which will detect the spectra of your sample. So remember, it's a destructive analysis. However, the minimum amount of the sample is really minimal. It's like from 1 to 10 milligrams, and 1 milligram, it's around 1/2 centimeter of a thread. So it's really not excessively, you do not have to sacrifice a lot of your textile, but also remember that image of the various concentration of dye in the fiber. So it's better if you can to have a little bit longer, like 1 centimeter, 1.5. This would be my advice if you want to analyze it. So so the column where these elements are separated are located in this machine, and this one is the mass spectrometer. And so in this small column, the various elements of your sample will be separated. And so the identification of the components consists in the comparison of the retention time of the sample signal and the standard. And then you will compare the absorption spectra or masses. In our case, we use a mass spectrometer. So the chemist will, so that's basically what will be produced. You will see this diagram with some peaks. And because the chemist had the standards, they can tell you these peaks at that frequency is significant of that and that component. So for example, in this small fragment of textile, woolen textile from Meroitic, we took a small red thread and the extract from this sample. So it was dissolved, and the result was that the sample contained alizarin and purpurine, which are in fact the markers of two possible dye sources, plant sources, madder or Our Lady bed straw. So this is the first answer we had. So our textile was dyed with madder or with Our Lady bed straw. And on that point, what we did is we were fortunate enough, because like Nubia say, like the natural dye plants are well studied by many people. And we have at our disposal, for example, this map, which shows you the areas where these big family plants are growing. So if we are talking now about a textile which exhibits all features of a local production, and we are located somewhere here in Lower Nubia, so it will be easier to make the choice between these two possible candidates and assess that it was madder which was most probably used to get this dark red color. And my next example, which will be like a transitional example between this dye identification and the next technique I will briefly discuss, it is provenance tracing. So this is a kilim. And this extraordinary piece was found in Meroitic in the layer dated to the 14th century. It was woven in wool, and after conservation, it appeared that all its width is preserved. It is actually 95 centimeters, and the actual preserved length is 1.50 meters. I'm giving you these measurements because I am aware that this image on the slide is not really, does not give you the real size. It is not so, it does not appear so big on this picture. So during the conservation process, so we were able to better understand how the decor was organized on it. And so if we add this killing technique and also the fiber spinning direction, which is not the traditional Nubian one, so we were pretty sure it was a textile made by foreign people. So when we crossed our sources and we discovered that the site was temporarily occupied by nomads from the Beni Inkrima tribe, we were almost sure that this was an imported textile. But we wanted to have additional proofs of this preliminary assessment. So we submitted a few threads of red, blue, green, and yellow to dye identification. So we did, so between the plant dyes, we had madder, world, reseda, luteola as well. But to our, what was also nice, it's that in almost all the samples, like the main dye source was an animal dye, namely from Kerialaka insect, which usually does not appear in locally produced Nubian textiles. In the analysis we conducted in my former project, Kerialaka was indeed identified, but almost in 95% in imported textiles. So this was for us a good signal that we are on the good track, and that the presence of this animal insect dye was for us an additional argument to state that this kilim was imported by this nomadic tribe from the Maghreb. And so we were very satisfied to see our theory very to be confirmed. And so we wanted to check also the provenance of the wool. So we will be 100% sure that the kilim was a foreign project. So a few words about provenance identification. So this is a method which is very effective, and it's used since many, many years in archaeology to trace mobility of human populations, of animals, but also provenance of various archaeological materials, including textiles. What is important, you need to have a good knowledge of the geological basement of the area you are working in, because various rocks contained or do not various ratios of this strontium 87. So this is important because it can influence the final ratio. And of course, it requires a knowledge of spatial variation of this strontium isotope composition. So it's good if you have if you can compare them with some earlier samples which could have been analyzed. As the HPLS-MS method, it's a descriptive method. And here, the weight of the sample is much more important, because you can see that the minimum weight is between 100 and 150 milligrams. And when we are talking about archaeological textiles, which are usually very dry and desiccated, it means it will be at least 3 to 4 square centimeters. So it is something you must keep in mind, and if you really have to choose carefully, because you will destroy much more than just a thread. And also before you take the decision to submit your sample and destroy those 4 square centimeters, which will be dissolved in the lab, you must be aware of the main important thing is that the question that strontium isotope analysis will answer to is your sample local or not? It means that you will have 2 answers. Yes, it is local, or no, it is not. But in the case the answer is no, it cannot necessarily give you the answer from where precisely was your textile imported. So be aware and do not be disappointed. I mean, it is something you must be aware if you want to trace the provenance, that perhaps your answer will be no, and you will have no clue from where it comes. Coming back to our kilim, so we actually, so this is which will be published soon. So this is these are diagrams prepared by my colleague Professor Belka from Adam Mickiewicz University in Poznań. And our sample, the kilim sample, is number 7 on that map. So you can see from where the textiles were retrieved. And we analyzed in that study both wool and cottons. And I only focus now on this sample number 7, which you can see. So we had at our disposal some results from strontium isotope signatures of these Capra remains found in the region between the 2nd and the 5th cataract. So these results were usually performed on material which was much more older than our textiles, but still it provides us a frame. And as you can see, our sample number 7, which is this, here, well, its isotopic, strontium isotopic signature aligns very well in this frame which were established. So it means that it is really highly probable that our kilim, which exhibits all features of an imported textile, was in fact produced in Nubia with a Nubian wool. So now we cannot rule out that the kilim was imported by this nomadic pastoral community from the Maghreb, from an area where the geological composition was similar to that of northern Sudan. But the most likely scenario is that it was produced from a local raw wool by the Beni Ikrima people during their stay in Mainarti. It, of course, opens various options. Was this wool collected from their own flocks? If so, it means they stayed at least one pasturing season to have the needed amount to process those fiber, dye them, and weave the kilim. We may not exclude that they use only the wool of local sheep. Anyway, I think it makes us aware of the many various possibilities I mean, before we assess very with a big certitude that something was important or not. So I think it was for us, it was a very big surprise. It was a reminder that to be very careful when we deliver such consideration about something which was local or not local. And it also shows that Nubia was, like from our perspective as archaeologists, to remember that Nubia was populated by various tribes, various cultural populations, and so let's still be careful when we have like our technical features and some certitudes we can have about. So as some final tips to at the end of the presentation, so I've reunited here so the basic information, including costs about those two techniques. So my idea was not to make you afraid and do not work with chemists. On the contrary, I think there are very curious people and we are curious people. But if we want to have a good work altogether, it's really important that both sides understand. We speak the same language, the same vocabulary, and so we will not be disappointed if we want how they work and what kind of results they can provide us with. And it's when we communicate with them and exchange that we can, I think, have a better knowledge of the context in which some textile we are studying were constructed, were created in. And I think I will thank you very much for your kind attention and hope I didn't lost you with all these technical terms. And I will be happy to answer any question or to make clear any things I might said which were not. Thank you very much.